And firstly, Omer Basha will present. He's from the Ben Gurion University of Negev. Is that right? Yeah. Okay.
Hi everyone, my name is Omer Basha, and I will present our work on differential protein interactions. So differential network analysis that highlights interactions changes between conditions is a rising paradigm in network biology, but differential network analysis is usually applied to compare between a few conditions, and a large-scale assessment of different methods has been lacking. So here we developed approaches that extend the paradigm towards differential network analysis of multiple conditions, which we applied to analyze protein interactions. Interactomes of 34 human tissues. So the notion of the differential approach can be summarized as the difference between the interactions in the tissue, for example, in heart, and the median interaction score across all tissues to give a score of how the interactions has changed in this context compared to all other contexts. Or if we want to be more formal, we can talk about it in the network notion. So in these networks, a high score means the interaction is highly likely based on the interactor's concentration in the tissue, and a negative low score means the interaction is less likely in the tissue. So interactions with a score close to 0 are not changed across tissues. And we've compared several weighing schemes to see which performs the best, and you're invited to my poster to hear more about them. So to assess the performance of these approaches, we wanted to check their ability to expose physiological differences among human tissues. So to this end, we manually curated 6,019 biological processes with their relevant tissues. Our analysis revealed the advantage of differential tissue interactomes over node-weighted node-weighted tissue interactomes in highlighting tissue processes. So as I've said, we tested several scoring methods, but due to time limitations, I will not present them here. And if you want to hear more about them, please stop by my poster. But what I do want to show here is that we're actually catching tissue-specific processes, as we can see in the diagonal of this bubble chart, representing the enrichment of tissue interactomes with tissue-associated processes. We're also seeing related tissues enriched for each other's processes, like different types of muscle tissues enriched together. We then tested whether differential tissue interactomes can highlight tissue-selective disease-related genes. So for this, we created another manually curated database of about 1,500 tissue-associated disease causal gene cases from the OMIM database. And after creating our tissue-associated disease causal genes dataset, we looked at the neighborhood of each causal gene up to its second neighbors, and we then limited our view only to the top 1% of the interactions in the related tissue differential network, and how many other causal genes with similar phenotypes are in the limited view. And we calculated a p-value for each of the disease cases. So in 25% of the cases we studied, the top 1% differential subnetwork surrounding causal gene was enriched significantly for additional disease-causing genes, a fraction much higher than expected by chance. And when we looked at specific tissues, we see that in some tissues we do better, such as muscle and heart. Such as muscle and heart. So to summarize, we've created two differential interaction approaches that capture tissue-associated processes and disease genes, and two data sets of tissue-associated biological processes and genetic diseases. Altogether, the extensive resources that we created in our analysis demonstrates that the differential analysis of human tissue interactomes is a powerful tool for highlighting processes and genes with tissue-specific functionality and impact. So all of these differential interactions data are available as a web database called DifferentialNet, and you can search for your protein of interest and view its neighbors. and which of its interactions are altered in the selected tissue, along with much more data about the proteins. So if you want to hear more about this work, please stop by my poster. The number is A220. Thank you.
Thank you, Omer.
Thank you.
I guess we skip questions again because we're a little bit limited in time, but